Good morning, everyone. I am just figuring out this video effort with my lectures and just information that I want to verbalize to you guys as we get through material. Right now, we should all be looking at Chapter 23, The Evolution of Populations. I'd like to go over the reading guide, questions 1 through 13 with you guys. I have the answers here. As time goes, I'll get these posted to your page so you can see them also, but for now, they're right here for us to go through together. As we look through number 1, evolution is a change in allelic frequencies in a population over time. What that really is telling us is that when we look at microevolution, we're really just looking at a gene pool and the number of dominant alleles compared to the number of recessive alleles for a certain trait. And if we see those frequencies or the amounts of each of those allele frequencies change over time, that is evolution occurring. And so that is that conversation I started last week with you guys in regards to microevolution. As we go down to number 2, the 3 main mechanisms that can cause these allele frequency changes from generation to generation. Well, we've already spoken natural selection. So if we're thinking in terms of chapter 22, yeah, there you go. Darwin and natural selection, the survival of the fittest or descent with modification over time. But as we look at B and C, there's some other mechanisms that can cause this change. Genetic drift, that's the one that's been a little tricky for folks. And I'll also talk about that in the lecture that I'll also post later on today over a PowerPoint. But as it says here, the variation in the relative frequency of different genotypes in a small population owing to the chance disappearance of particular genes as individuals die or do not reproduce. This is not a natural selection thing. It just means that in smaller populations, frequencies can change very quickly if individuals just die that have nothing connected to survival of the fittest. Due to chance loss of particular genes, that will change frequencies, which will show a response of evolution. Gene flow is the next one, which is just individuals moving in or moving out of a population can effectively change the overall allele frequencies of that population. As we go to here, sorry, I'm just kind of playing around with a couple things. I don't want to play with that right now because that's kind of not great. I think otherwise we're good. Moving on down to number 3, when Charles Darwin wrote On the Origin of Species back in the 1830s and 40s, he knew nothing about genetics. Remember I mentioned last week that Mendel's work wasn't done until the 1850s. And so Darwin did all this blind without a knowledge of genetics, which makes it incredible that his work still stands to this day. And actually the work of Mendel and the genetics work that we've learned over the last 150, 170 years has only given more solid foundation to Darwin's work. Number 4, genetic variation is the differences among individuals in the composition of their genes or other DNA sequences. We talked variation with our opening lab for evolution, and that is that base piece. There has to be genetic variation in a population that will allow for change to happen in that population over time as environmental pressures change. Number 5, many phenotypic characters are influenced by multiple genes, like coat color in horses, seed color in corn, and height in humans. We spoke about this back in chapters 14 and 15 when we spoke about polygenic inheritance. Many genes can influence a certain trait. As we go to number 6, genetic variation, that is that raw material for evolutionary change. Without it, evolution cannot occur. Number 7, new alleles. New things arise in our DNA through mutation. Mutations that occur to our DNA, as much as we may think of them in terms of just being negative things, mutation carries a negative connotation unless you're a big X-Files fan and want to be, I said X-Files, let's try that again. You know what I'm talking about, the X-Men. The X-Men, the mutations they get could be very positive, but when we think about mutation in humans, we think about negative consequences. But we want to keep in mind that there can also be just the raw material for change. And changes in our DNA can lead to new phenotypes, new things. Moving on, number 8, how can harmful alleles be hidden in diploid organisms? Heterozygotes. If the harmful allele is recessive, someone can be a carrier, and that allele still survives. Number 9, are all mutations harmful? Of course not, as I mentioned before. Much of our DNA is not even coding regions for proteins. It's all that in-between stuff. And so if there's a mutation in a non-coding region, it actually isn't going to affect a protein or the expression of that protein, and we move along like normal. The second bullet point says there's many silent mutations because of the redundancy of the codon chart. Remember, we learned in chapter 17 about protein synthesis, we learned that many different codons can code for the exact same amino acid. So if there's a mutation that causes the same amino acid to be produced in the ribosome, then there's no change. Number 10, how can a gene translocation? A gene translocation, we haven't learned that term, and I'll actually add to this later just to give this a little bit more, hopefully a little more understanding to you guys. But a gene translocation is on a chromosome where a piece of a chromosome actually, through the process of replication, actually gets put onto the wrong chromosome. And what that can lead to for organisms, this doesn't happen very often in humans, but this can link genes in new ways and connect them in ways that actually can create positive effects of survival. And I'll do a little more digging also to see if I can give us a specific example to help with that. Number 11, do eukaryotes or prokaryotes have a higher rate of mutation? Who has more generations per unit of time? Well, this comes down to the fact that eukaryotes, our cells, we have a higher rate of mutation, which this number is just good, just good common trivia you can share with your buddies. But our rate of mutation is still low at 1 in every 100,000 genes per generation. So that's super low. Prokaryotes have a lower rate of mutation, but they reproduce a lot faster, as we learned when we did our pGlow lab a month ago, where they go through a lot of generations very, very quickly. So over time, prokaryotes will develop more variation in their population, genetically speaking, in a much shorter period of time. Number 12, for organisms that reproduce sexually, most of the genetic variation in a population results in the alleles an individual receives from its parents, which, well, makes sense. But that's really what we want to start to understand is the importance of sexual reproduction to creating genetic variation in a population. If we think about bacteria very often, when they create their next generation, they make a copy of their DNA and then they split into 2 cells. Well, there's no genetic variation being created there other than just the mutations that are occurring, and that's just random happenstance that's going to affect a gene of interest. With sexual reproduction, if we go down to 13, sexual reproduction shuffles all the existing alleles in the mother and the father, for instance, and deals them randomly to the next generation. This contributes to variation in a population. So things like crossing over in meiosis, that can lead to new combinations of alleles in the genes of the gametes, the sex cells. Independent assortment of the chromosomes. We learned about this term when we learned in meiosis a little bit, and then also in genetics about just how the chromosomes line up in meiosis can lead to different alleles in the gametes. And so the quick connection we can make to that is just the connection of ourselves to our siblings and to our recent or our most closely related family members. We have the same genes coming from the same parents, but there's a lot of different combinations that can occur through, again, crossing over in meiosis, creating some new combinations there, how the chromosomes line up in meiosis, that independent assortment of the different chromosomes, the 46 different chromosomes that we have, and finally, just which sperm cell fertilizes which egg cell. If we remember, again, through meiosis, there's 4 different cells created, 4 egg cells, 4 sperm cells created for each attempt at meiosis. So there's a lot of different possibilities of which could fertilize which to lead to variation. And so I'm going to leave it at that. And I'm going to post this a little bit later on if I can get things worked out right. And then I'm going to follow up. You'll also see a post of the PowerPoint lecture. The PowerPoint is already posted on your page for today. You're welcome to go through that. I'm going to work to jam through that here in a few and get that posted later on. Thanks so much, guys. I hope you're doing well. Give me a shout if you need anything. Take care.